This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 78 for April 16th, 2010. Hey everybody, I'm Vincent Racaniello, and this is TWiV, the show where we talk all about viral videos. No, that's the wrong podcast. What do we talk about on this show, Dickson?
Talk about viruses, the kind that make you sick.
Or might not.
Well, that have the potential for making us sick.
And joining me today to do that is Dickson Despommier.
Hello, Vince.
Welcome back. We missed you for a week.
I felt the absence myself.
You were at the UN.
I was.
What were you doing there?
I was talking at a symposium dedicated to improving the world's water supplies.
Uh-huh. Perhaps that's where the Latin phrase has come from in our show notes.
Perhaps.
So also joining us today from Western Massachusetts, where there's plenty of water, Alan Dove.
Yes, there certainly is. Good to be here.
Is it raining today?
No, we're supposed to get more tonight, though.
Uh-huh.
But apparently we're not going to be measuring this one in feet, at least.
This is good.
And finally, but not least, Richard Condit from Florida.
Hello. It's good to be here again.
Would you say you're in Central Florida or Northern Florida?
We call this North Central Florida.
North Central, I see.
Way to hedge your bets.
That's right.
At the hinge of the panhandle.
There is a Latin quotation on our show notes. Dickson wanted to know how to say in Latin, water is too valuable to use only once.
Or to drink.
I don't know if it's correct. I found a online translator. Why do you want to know it in Latin, by the way?
Well, I want to put that on the first vertical farm.
The first one that's built?
Yep.
Oh, that should be nice.
Yep.
If you're still alive. Oh, sorry.
Thanks a lot,
Not
Vince.
very nice.
Well, actually, Dick, I meant to forward you something earlier today, and
yes.
I didn't get to it. It was a press release about a company that's building hydroponic systems inside tractor trailers.
Oh, nice.
Although their intent is for a crop that I don't think you were planning on growing.
Right. High return on investment, but it's got a lot of risk.
So what would you call that?
Unless it's in California.
The horizontal farm.
Yes, actually, they were calling it the Big Bud.
Yeah, well, there's a lot of that out there. But
it's ostensibly it's only for medical purposes.
Oh, but of course.
Get the real thing. Get the Dickson Despommier approved vertical farm.
That isn't even vertical anyway.
Yes, it's not. Anyway, we have as usual.
Wouldn't be for long.
Yeah, we have as usual interesting virus stories, which we don't dumb down. We keep them pretty technical.
We do.
And we don't charge you for them either.
I'm the dummy that asks the stupid questions.
No, you're not a dummy. We don't charge you. This is free because knowledge should be free.
So, Vince, start the show. I thought we were going to start the show with a joke.
Oh, we made up a virus joke.
Oh, you did. I thought it was a very
Do
clever
I have
joke.
to take credit for
I
this?
would
All right.
love to give you credit for this joke.
Two viruses walk into a bar. The bartender looks at them and says, "Are you sure you two are old enough to drink?" And one of the viruses says, "Yeah, I'm positive." It's not done yet.
Wait, there's more.
No, that's it. There's more?
Yeah, there's more. Then the bartender says, "I don't believe you." And they were stranded. Yeah, it's pretty bad, isn't it?
Did you really make that up?
We did, actually.
Yeah.
We conspired that one.
I thought the positive part was good.
You're going to get email about that.
That's all right. Let's do some positive thinking about viruses. Actually, I wanted to say one more thing before we talk about viruses. We provide this podcast for free because I think knowledge should be free. And someone asked Jonas Salk, the developer of the polio vaccine, why he didn't patent the vaccine. Do you know what his answer was?
That would be like patenting the sun.
Exactly.
Aha.
So knowledge is similar, isn't it?
Tell that to Craig Venter.
What does he have to do with it?
He patented the entire genome of the human.
All right. Well, that's a discussion for another podcast. We have 3 cool stories today. And the first one is about a way to treat arthritis with a protein from a pox virus, which sounds like it's right up Rich Condit's alley.
Right up my alley. Actually, it's better up Grant McFadden's alley, who also works here. This is his story, actually.
And he's up the hall from you.
He is, he's upstairs. Actually, he's not upstairs right now. He's in Maryland or something. He's on the road a lot. But I actually had an opportunity to I talked with him for about an hour about this story. It's a great story. So I guess this starts with Vincent, you finding this. Do you call this a press release or something?
I don't know what this is. It
Whatever
looks like
it is.
it looks like a press release of some
Yeah.
kind.
From
Viron.
I think it's from it's from Viron Therapeutics.
Yeah.
Yes, this is a press release.
Okay. And it's talking about this anti-TNF protein therapeutic called VT346 that is being developed to treat inflammatory diseases of one sort or another. Specifically, they talk here about arthritis. But the fact is that anti-TNF molecules can be used to treat a variety of diseases. There's one on the market, Remicade, that's used for psoriasis, Crohn's disease, ankylosing spondylitis, psoriatic arthritis, rheumatoid arthritis, and ulcerative colitis. So this is a family of diseases that are autoimmune diseases where you're reacting immunologically against yourself, and there's a whole bunch of inflammation. And key to that is, in many of these diseases, an inflammatory molecule that we make that's part of our cells called tumor necrosis factor.
Right.
A protein that is a cytokine that regulates a lot of other cytokines. Cytokines are signaling molecules that regulate the immune response. So apparently TNF is central in arthritis, for example, and there's been a lot of research to suggest or to show that if in a person who has rheumatoid arthritis, if you can somehow counteract or remove the tumor necrosis factor, that it has therapeutic value for the arthritis. Right. So there's a huge demand for this kind of thing.
And this is also, I should just point out that tumor necrosis factor, people who are listening might think, well, gee, don't you want something that kills tumors floating around in your bloodstream? But it turns out that's just an accident of the way it was initially discovered, right?
Right.
This turns out to be a very bad molecule to have floating around in large quantities.
Right. Yeah. Enough at the right time is good.
Yep.
Too much at the wrong time is bad.
So we probably could say that whenever you get a virus infection, very early on you make a little TNF, and that's why you get fever and redness and soreness and pain. That's all due to TNF in part.
TNF and interferon and that kind of stuff. When I get a cold, my body feels just awful like that. I'm encouraged because I know my body's doing the right thing.
Exactly.
That's good. So I put a positive spin on that.
Yeah.
So at any rate, there's a huge market for things that will somehow get rid of the excess TNF involved in an autoimmune disease. And this press release has to do with manufacture of one such molecule that it turns out is made by a pox virus called tanapox. So let's talk about pox viruses and tanapox and immunomodulation a little bit. So obviously, when a virus infects a cell, the cell mounts an immune response. A lot of viruses have figured out ways to combat the immune response by making what we call generally immunomodulatory molecules. And the pox viruses are champions at this. It's a double-stranded DNA genome. They encode about 200 genes. They're big viruses. And I'll bet you easily 30% or maybe 50% of the genome is devoted to making immunomodulatory proteins of one sort or another that bind to and inactivate all sorts of cytokines and other inflammatory molecules and do all sorts of other tricky things to modulate the immune response. And people have, over the years, once understanding this, have sort of mined pox viruses for various immunomodulators. And there was a guy named Karim Asani, who was studying tanapox virus and noticed a cytokine binding protein that was secreted from infected cells and apparently had activity towards lots of different cytokines. and having a little difficulty sorting it out, and so enlisted the help of Grant McFadden, who had, in fact, just around that time, finished the sequence of the tanapox genome. And Asani and McFadden and his lab together purified this protein. I think what Grant said was they made a TNF column and poured supernatants from tanapox-infected cells over it.
Nice.
Sounds like a good way to go.
That's good.
So by affinity chromatography, pulled out this molecule and got a little amino acid sequence from it. And since they had the sequence of the genome, they were able to identify the viral protein that was involved. And the interesting thing is that it looked nothing like what they might have expected. There's lots of pox viruses that make TNF binding proteins, and most of those TNF binding proteins kind of look like TNF receptors. Okay, so TNF exerts its action ordinarily by binding to a cellular receptor.
Yep.
The virus makes proteins that are soluble like TNF receptors, so they go out and sop up all of the TNF and thereby inactivate it, keep it from doing what it's supposed to be doing. Well, this thing doesn't look like a TNF receptor. It looks like an MHC1 family molecule. And why that is, Grant doesn't know. Nobody knows at this point, but it's a little quirk of evolution.
So MHCs are major histocompatibility proteins.
Major histocompatibility molecules also involved in the immune response for T and B cells recognizing each other and antigen and triggering the whole immune response. So
Rich, can I interrupt you just a moment?
Sure.
What does this virus infect in nature?
Thank you for asking.
You're welcome.
We don't know.
Well, how did you isolate it?
So
where did it come from?
It shows up in humans. Kind of like I've never seen a tanapox infection, but it sounds kind of like a cowpox infection, where you get like a single isolated blistering lesion that heals and then you're okay.
Yeah.
It's found in Africa. It is one of several genera of pox viruses.
Got it.
So there are vertebrate, a subfamily of pox viruses are the vertebrate pox viruses, and then there are 1, 2, 3, 4, 5, 6, 7 different genera, 8 different genera, including the Yatta pox viruses. And within those is the species tanapox virus. There's only 3 viruses in that whole genera, all from Africa. The guess is they probably have a rodent host like so many other pox viruses, but they don't know what it is. There's some suggestion because of the appearance of these isolated lesions with no other explanation that maybe there's an insect that vectors them to humans, but there's no proof for that.
Why is it called Tana? Because it's from Tanzania?
You got me on that one.
That's from...
It's right.
It's a
I was
location.
just looking at it. Yeah, it's
most of them are named after places, right?
It is named after a place, and I was just the Tana River Valley of Kenya.
Kenya.
Okay,
there you go.
The next country.
Right.
Got it.
All right.
Yeah.
Cool.
So, so they isolated this molecule, and it has an affinity for TNF that is, my understanding is that it's orders of magnitude higher than the best TNF binders out there on the market.
Son of a gun. So
this has, theoretically at least, very significant potential as a therapeutic for autoimmune disease and specifically arthritis.
Very nice.
So this is being produced by this company Viron, which in this press release says Viron was established by 2 Canadian scientists working at the Robarts Institute. Those scientists are, guess who? Grant McFadden and his wife, Alexandra Lucas. And there's an interesting backstory to this as well, because the company was founded on yet another pox virus anti-inflammatory molecule, this one from Myxoma, called SERP1. And this is an interesting sort of semi-serendipitous story, because Grant is a pox virologist, his wife is a cardiologist. This SERP1 is it's a serpin, serine protease inhibitor. And these protease inhibitors are important in regulating the immune response as well. And basically, when Grant discovered this molecule, he collaborated with his wife to show that the anti-inflammatory properties of this molecule would, in fact, prevent restenosis of atherosclerotic lesions after angioplasty. A big problem in atherosclerosis and treatment is you blow these things away with a balloon catheter, but then they come back, they restenose. Of course. But if you infuse this anti-inflammatory molecule at the same time, you can retard or prevent the restenosis.
I think that's why they recommend aspirin therapy too, right? To keep down the inflammation so that you don't get these clots forming.
So at any rate, that molecule has made it through phase 2 trials, and they're looking around for somebody to sponsor a phase 3 trial. And this new molecule, the tanapox molecule, the TNF inhibitor, they're ready to start clinical trials on that. And this press release, I think, is, among other things, a fishing expedition saying, look, we have an investment here, someone come and help us run a clinical trial.
Sure.
Right, and companies, by the way, are also legally obligated to make public announcements about major research findings if they could affect the stock price.
Okay.
So one reason you'll see these press releases and you think, well, gosh, this isn't really something anybody's ever going to write a news story about. And it's for legal reasons. They have to make an announcement, and the cheapest and easiest way to do that in many cases is through a press release. But yes, of course, they want to drum up investment for this thing because now it's ready to go to the clinic.
Right. And actually, when I talked to Grant about this, I said, well, geez, you ought to be telling this story. And he says, no, you should be telling this story because, because I can't just go and tell the whole story. You know, I can get into all sorts of conflict trouble.
Yeah. Oh, that's great. Another great example of how a virus or part of a virus might help us.
Yes.
Yep.
Right?
You bet.
And
turning their swords into our plowshares.
I think it's great.
Or put another way, we're learning to think like viruses.
Hear, hear.
Well, you know, this is another reason you just study a virus. This is an odd isolate from somewhere. You find something new. It just goes to show you have to just be curious.
But remember what Louis Pasteur said.
Curious prepares the lucky mind. No. luck favors the prepared mind. That was
probably, that's more like it, Vince.
Luck favors the prepared mind, yes.
That's right.
Well, we're all prepared. We're just...
You got to know when you've been lucky.
Yeah. This is great. I love this story. I'm looking forward to talking with Grant when I visit Florida next month.
You'll have a chance. You'll have a good time with him. He's right on the money, very articulate.
We're going to do a TWiV, and then he can talk about something else that's not proprietary. And he has many interests, one of which he'd probably like to talk about is the use of pox viruses as oncolytic agents, anti-cancer agents.
Right.
Okay.
All right. Perhaps we'll do that. Okay, number 1. Number 2 is a story I found a couple of weeks ago, and it is called Darwinian Evolution of Prions in Cell Culture. Now how can a protein evolve? Caught my eye. It's from Charles Weissman's lab. Now, Charles Weissman, if anyone remembers, used to be in Switzerland. He's quite an accomplished scientist. And he has recently moved to Scripps, Florida. Scripps started a new branch down there. Is Jupiter, Florida? Is that near you?
No, that's on the
coast.
On the Atlantic coast, significantly south of here.
Anyway, he's in the Department of Infectology.
Yeah, I thought that was pretty interesting. Where did that come from?
I don't know where that came from. Maybe he made it up. Anyway, so he's doing prion work. And so this paper is very cool. Prions, everyone may remember, are proteins that cause some diseases, although not all prions turn out to be causing disease. Scrapie, for example, BSE, Creutzfeldt-Jakob, they're all prion diseases. But in yeast, prions do good things. They're basically proteins that make other proteins look like them, make other proteins fold like they do.
They make a good impression on them.
Excellent. Very good. They make a good impression on other proteins. So many people have been studying sheep, prions, and various others, and you can do them in mice and you can do them in cell culture. And this is what they do in this paper. The neat thing is you can take, say, a mouse with a prion disease, which is a neurological disease. It's a degenerative neurological disease. You can make an extract of their brain and put it into certain kinds of cells, and the prion protein will be taken up and grow and multiply. It will convert the prions in the cell, the normal prions in the cell, into pathogenic prions. And as you split the cells, they will stay in the cell and be perpetuated for many generations of cells. So you can put them in cells, or you can put them in animals. If you take a pathogenic prion into a healthy mouse, the mouse will develop a prion disease because the pathogenic protein converts the normal mouse version of the protein into a misfolded protein, or not misfolded, but differently folded.
This cell culture thing was news to me. I had no idea that they could propagate prions in culture.
Yes.
That's really amazing.
It's very cool. And it turns out that you can define strains of prions based on what kind of animals they infect, what kind of diseases they cause, the incubation period, and what kind of cell lines. There are a couple of cell lines they use for these experiments. And there's a pattern of infectability among these different prions. So there are strains. And in this paper, they use the cell lines to look at evolution. And I think there are two things that they do. One is if they take a prion and put it into a mouse, it causes disease after so many days. And you can take that prion out and you can put it in cell culture. And you can show that it changes as you switch hosts. And they show it changes in two ways, by the kinds of cells that you can infect. So the mouse-grown prions will infect a certain number of cell lines, and the cell culture-grown prions will infect a different pattern of cell lines.
Can I interrupt you just a moment
Of
here
course.
to come back to something basic?
Mm-hmm.
And that is that everyone out there that's listening to this says, wait a minute, DNA makes protein.
Right.
And then proteins regulate the formation
Okay.
of DNA. And I understand that part, but how can a protein that's foreign to the cell end up making the cell produce another protein like itself that it could then perpetuate?
Well, it doesn't. It just takes the protein that's already in the cell and causes it to fold in a different way.
So that's got a completely different amino acid sequence.
No, it's the same amino acid sequence.
It's the same.
Highly conserved.
Highly conserved. It may be one, depending, there are variants that have one or two amino acid
But
differences.
folded in one way, it does something good. Folded in another way, that this
Exactly.
other one says does something bad.
The
And
folding
the
is quite
misfolding
different.
can be templated by the misfolded protein.
Got it. But then that's perpetuated to the next generation?
Yes. And the thing is that when you, for example, go from a mouse to a cell culture and you change the strain, you're not changing the protein. You're changing the way it's folding.
Right.
So all the differences we're talking about today are all folding differences. Maybe the one that's the easiest for us to grasp is they use a drug. They use a drug that's an inhibitor of the addition of sugars to proteins. It's a glycosylation inhibitor. Certain strains of prions are sensitive to this drug
as well they should
in
be.
cells, and others are resistant. Okay, and so those two proteins are exactly the same sequence, and so they say, well, they must be folded differently. How could they have such different biological properties? So they're doing a series of experiments where they found that this one prion in a cell was sensitive to the drug, which is called SWA. And so they said, well, let's try and cure the cells of the prion by treating it with this drug. That could have interesting consequences for treating people, you can imagine.
Yep.
So what they found is that after a few days of treatment, the prion levels went down. Then all of a sudden they went back up again. And then all the prions in that cell were resistant to the drug. But they're the same protein, so they must be differently folded. How differently, no one knows, but they're simply differently folded. And then they do some other experiments to ask whether you can do you select for pre-existing resistant prions, or do you actually make them resistant?
Goodness.
So the answer is both, depending on what you use as starting material. Sometimes if you take a cell culture grown prion and you add drug, they've shown that there's a little bit of about 0.5% of the prions made by this one cell line are already resistant to the drug. But then they cloned the prion. They did limit dilutions to make sure there's only, say, one prion being inoculated, and then they still get drug resistance. So the drug can push the misfolding of the protein. They can select for it. And so they actually, in other words, the resistant variants arise during the propagation of this limit dilution cloned prion.
So, but I'm still a little bit worried about the use of Darwinian selection here, because that's obviously a genetic thing, and this is not a genetic thing.
Why do you think it's genetic?
There's no DNA involved in this.
Darwin didn't know about DNA.
No, I know that,
Yeah.
but it has come to be known as that, gentlemen.
But it's
When you say Darwinian evolution, you can talk about how the genes behave, like Mendelian genetics and all this other
But
stuff.
there is apparently a heritable factor here that is being passed down from one generation of prions to another.
Right.
And in
No,
terms of this sort of mutation-like phenomenon and the selection, it's behaving in a Darwinian fashion.
Right.
So-
So this is setting a precedence for the use of the word
Exact.
Darwinian.
Yes, absolutely.
So
It's
this is a landmark paper.
it's
a landmark.
it bends your head. For
That's right.
sure.
Yeah.
Yeah, because it's totally unexpected.
Well,
Out of
and it
left
tells you
field.
that evolution as a process can occur anytime information is being transmitted through
Yeah.
a heritable system, and it doesn't matter what the what the data storage
No,
method
Alan,
is.
that's a great point.
So they
It's a brilliant question.
they compare it to heterogeneity, like for RNA viruses, and they say although heterogeneity in the case of RNA viruses is due to point mutations resulting from error-prone replication, heterogeneity in the prions is likely to be due to differences in the structure other than the amino acid sequence of the prion molecules. So this is something new. But they also say maybe there's a small RNA associated with these that's involved because maybe you couldn't detect it.
Yeah.
Or a variation could be due to cell-derived determinants, for example, association of the protein with a small cellular RNA. But I like the idea that it's the structure of the protein. Now, we don't
So
know.
do you know whether there are really good three-dimensional structures of like some of these active and inactive forms, and whether this templating and conformational change in any case is really understood mechanistically? I mean, I don't know the answer
No.
to this question.
There are some structures, so we know that the pathogenic protein has a lot of beta sheet, and the normal cellular protein does not. But we don't know how they convert.
Right.
How that happens mechanistically is completely
Someday
unknown.
we will,
I'm
Indeed.
sure someday we will, but at the moment we don't.
because
Anyway,
this really challenges my imagination. I think probably
you,
because
Rich.
I'm so sort of DNA based.
Yeah. No, that's
I'm
exactly
still a little
what I
squeamish about this, but it's amazing.
I think that that's a good point to have brought up, because that's what we're emphasizing, that this is the non-DNA based inheritance. And similar things happen in yeast. And in fact, Susan Lindquist's lab at MIT works on these yeast prions, and has also said that it's a Mendelian, non-Mendelian inheritance because it doesn't involve
Right.
DNA.
Okay.
But it's Darwinian because it involves selection.
Yeah.
Right? That's the
Okay,
key.
so there's a distinction. That's good.
And
Okay.
I think that's
Okay.
really cool.
Actually, I think that the non-Mendelian part probably is because it doesn't involve mating.
You think?
Or it doesn't select out 1
Right, 'cause
to
they're-
2 to 1 or something simple. like
Yeah, because Mendel
right.
didn't know about DNA either.
that. No.
Okay. But we know that involves...
But in retrospect, we could explain everything by DNA that he discovered.
well, he could explain it by having 2 copies of each gene, right?
Right. He was
correct.
doing crosses of
Correct.
things and
Right.
getting
Correct.
the Mendelian inheritance from that. So I
Exactly
think
right.
this is non-Mendelian in the sense that it's asexual.
Right.
Wild stuff.
This is cool. This is very cool. Anyway, that's Charles Weissman. And Rich, man, you found this webpage,
Isn't that great?
which is so cool. It's it's at the University of Zurich, which is where he used to be. And he has a little essay about some thoughts on the pursuit of success in science.
Yeah, I thought that ought to go on the
We're going to
on the
put this in.
website because it's really nice. It's really nice.
I want to read a couple of things. He says, only a chosen few achieve scientific breakthroughs which result in fame, but all of us have the opportunity of enjoying the excitement of a successful experiment and collecting the pebbles of facts which are ultimately integrated into the edifice of knowledge. I'm a pebble collector, Dickson.
If you're a pebble collector, I can use the sand as the mortar that goes in between the grains.
I also like Saul Spiegelman commented, one result, one paper. He said, you get a result, you write one paper. He also observed that anything worth publishing once is worth publishing many times.
He's got nice guidelines for giving a talk.
Those are
Right,
great.
Right.
Yeah.
If you don't understand a question from the audience, start out by saying, "This is a brilliant question," after which the questioner won't care very much about your answer.
This is all true.
And he's got a quote from the Talmud.
Read that, Rich.
If you pursue fame, it will flee from you. But if you flee from fame, it will follow you. And he says, to this my father added, but don't run too fast.
That is great.
It's good stuff.
Yeah, he's a very
entertaining man.
Yes.
Yeah.
Anyway, we'll put that in for sure. Okay. And before we move on to number 3, I'd like to acknowledge the support of Data Robotics of TWiV. Been supporting us for a couple of months now. We appreciate it. And they, of course, are the makers of Drobo, the storage solution for your computer. RAID made easy. Now, Drobo, if you go to drobo.com/TWiV, you will see they have many models with different numbers of hard drives that you can put in them. And one that just caught my eye is brand new. It's called the Drobo FS. And I believe it's a 5-drive model. So these Drobos are containers for hard drives, and they have slots that you slide hard drives in. You can put in 2 or 3 or 4 or 5 or more, depending on the model. And the neat thing is you copy your data to these drives, and it's protected. If a drive fails, you just slide it out and put a new one in, and it's restored from the remainder of the drives. They have a Drobo FS, which is brand new, which you plug into your network, and it can be accessible to all the other computers on your network. Now, Dick is blinking because you don't have a network, right?
That's correct. I have a neural network, but it doesn't work very well.
That's not true. You deal with the sand, yeah.
I'll take the sand. Goes right through my fingers.
But if we have a network here. If I plug this Drobo FS here, my lab could get it very easily. At home, I have a little network, and you guys probably all have networks at home of some sort.
Yeah.
You have a sneaker network, Rich?
I have a lame network. I need to reboot the whole thing because it's confused.
Anyway, if you have a network, you can plug this thing by an Ethernet plug, and it's available to everyone. And then wirelessly too, it would be available. Anyone that has a wireless connection could access all your data, so it could all be shared. So it's a very cool product. Go over there and check it out, and you can get some good money off of Drobos if you buy them empty or with drives in them. If you use the coupon code TWiVPod in capital letters. And as you know, we're in the process of giving away a couple of Drobos. We've given away 2 so far, and the third contest is coming up. The Drobo is going to provide us with the details in a week or so, and then we'll announce them here. Great product. Check it out. I have 3 of them. Anybody has a lab or valuable files, you should try it. It's a really good product. I endorse it fully. And we thank Drobo, of course, for their support of This Week in Virology and for making education and knowledge free.
That's the whole idea.
Did you get in touch with our second winner, Jeff? Was he...
Jeff was very happy.
Good.
I got in touch with him. He's getting his Drobo, and I said, in 6 months, write us a letter and tell us how you use it, and we'll tell people on the show.
Great.
All right, number 3 is a story discovered by Alan, but which Dickson Despommier will like.
Yes.
I like them all so far. Why shouldn't I like this one?
I think this one will really catch your attention.
Well,
Nick.
okay then.
So this is a group in Europe primarily. Actually, I guess there are several institutions. They're at Princeton and also in the Netherlands. And they looked at H5N1 outbreaks, bird flu outbreaks in Europe for 2005 to 2006, and obviously a lot of people have looked at that, but these folks wanted to know if there was any link between the bird flu outbreaks in Europe that year and the movements of wild birds.
You're talking about outbreaks in birds, right?
Yes.
Not
bird to bird.
Right.
Yeah.
Right. Or wait, no.
Because there are only sporadic cases of
Yes, there
this is outbreaks in birds.
Right.
in birds. Yeah.
This is outbreaks in birds.
Right.
So these are outbreaks in local birds.
Yes.
Right. Domestic.
Correct. And
very important.
And not in the Cockney sense of that word.
Not in the wild birds that they were looking at the movements of. So
That's right.
migratory birds have a little bit of a problem. They have to get far enough into the warm area that there's water, particularly for water birds. They need to be able to land on it and feed in it and all birds, of course, need to drink it. But they don't want to stray too far from their northern destination because then they have to expend a lot of energy to get back. So it turns out wild birds tend to, these migratory birds tend to spend a lot of time right on the 0 degree Celsius isotherm, right in the area. If you looked at a weather map where it's transitioning from freezing to thawing or thawing to freezing. Right. So these folks looked at the pattern of cold fronts in Europe during the 05 to 06 winter, and they found that, not surprisingly, the wild bird counts were high right along the isotherm, right along the 0 degree line. And interestingly, H5N1 outbreaks tended to occur in areas where surface temperatures were freezing to just slightly above freezing on the 2 days before the outbreak.
Right.
So there's a weather link to outbreaks of H5N1,
Got it.
which suggests that you could maybe predict these things.
You betcha.
Really interesting. I like a couple of things I like about this. I like just visually the idea that the isotherm concentrates the birds along that isotherm.
Yeah.
And I also like the idea that, there's these people out there counting birds the time.
Yeah. All the time. That's exactly right.
Yep.
And that's nothing to do with H5N1.
Yeah, and you wonder, why are these people counting birds?
That's right. Or how can I use the data?
Well, they're also looking at infections with H5N1 too, right?
Yeah.
Well, they've got their own reasons for doing this. There's all kinds of reasons for doing it, but all of this comes together in a fashion. The meteorology, the counting of the birds, the infectious disease stuff, all comes together to explain an epidemiological pattern of the infectious disease. It's great.
Fabulous.
Yeah, the bird counts actually were not done by these researchers. They didn't go out there and sit in the cold and count the birds.
Someone else did that.
Those data were already available.
Right.
By people who count birds because they want to know where the birds go.
Exactly.
So then explain this figure 2. The y-axis is in frequency of outbreak location, and then on the x is the surface air temperature. And it's called a significant association of outbreaks of H5N1 infection in wild birds.
It's right between the 0 and
but they say wild birds. So are they looking in wild birds here? They must be, right?
Yes, I think so. Yes.
Yes.
So this is in wild birds like mallard ducks.
Right. So they're looking, I'm sorry, I got it exactly backward. They're looking at transmission from the poultry to the wild birds, I guess.
Well, just transmission of the H5 among wild birds.
Right.
So I think the idea is that when these guys move in big numbers because they have to get to the water that's not frozen, they bring the virus with them and transmit it to other birds. And that's when you see these outbreaks.
That's right.
So I guess...
Trying to stay as close as they possibly can to their northern range, okay,
and still have
still have water. So they're concentrated along this front, and where they're concentrated, there's a high enough number to promote the transmission of the virus.
All right, so I'll be a devil's advocate. This is an observational study.
It is.
Right?
It is.
So maybe there is something else that we don't know about that is really doing this. It's not the temperature. It looks like it's the temperature because that happens to correlate well, but maybe it has nothing to do with it. Maybe it's some other factor that we don't know about, right? Isn't that possible?
Associated with the isotherm, but not because of the isotherm.
Right.
Because there's a 100% correlation between that temperature zone and those birds being there.
Right, but it doesn't mean that it's the temperature.
Yeah, you're right.
On any given day, the sale of ice cream cones is directly proportional to the number of drownings. Okay, so does ice cream cause you to drown?
That's right.
I mean, Dickson, if I get up in the morning and tie my shoes and then it rains, does that mean that...
No, of course not.
So something else could be, and I know that when you do these observational studies, you can eliminate some other confounding things, but I don't know if they've done that for everything.
So here's the question to all of you then. What's the test for this one? How could you do a laboratory simulation of this and get it
lab simulation is tough.
I know you do. It's very difficult,
This is
right?
an ecological issue, right?
Of course.
But maybe you could do something outside. You could do a random, double-blind, placebo-controlled... I'm just joking.
No, what you could do is have flocks of birds. I mean, these wild birds are things like mallards that you could you
Right.
can you can grow in flocks
Right.
at home. So if you had varying size flocks, you could look at transmission rates of the virus within those flocks based on flock size,
Yeah. Well,
which would give you. I mean, that's another. It would not necessarily be a lock on this being,
no.
but it would be another relevant
Yeah.
data point.
The biggest difficulty with ecological studies in general is that they are all observational to begin with.
Yes.
And then you perturb half and leave the other half the same, but it's not an equal half, and it's never the same environment for both of the areas that you study. So it's very difficult to do hardcore science on the environment. Very
Now,
So
if you could get all these bird counters to go out and round up the birds and bust them some,
I was gonna do the opposite. Get the bird counters to go out and shoo all the birds away.
Yeah, right.
And disperse them.
Yeah, sure.
Well, look, even if it's another factor, if it always goes with the isotherm, the 0-degree isotherm, then you could follow that and see if, in fact, you can predict outbreaks.
Which is what they did, and I guess that's what this data is based on.
Well, this is retrospective, right?
Yeah.
But now you would like to do it actively
No, that's right.
in the winter and
I mean,
see.
the same problem arose in accounting for the spread of West Nile from the east coast to the west coast of the United States. And if you look at the data, it all seems to follow the major highway systems moving
But what do you
from east
do?
to west.
But what do you do, Dickson?
Well, then someone released a genetic strain of mosquito to prove that indeed they could move that fast from east to west.
All right, so you want to release altered birds?
I don't want to do that.
I don't think we should.
I don't know
that.
what the experiment is here, to be honest.
Well, if not the experiment, how do we use this information to prevent, outbreaks? Of
I need
H5N1?
5 years in a row of that observation, and I need to say every year that it was exactly what it was the first year I observed it.
That's
once you've
true.
got that kind of data behind you, then I think you've got some power.
All right, let's say we have those data. 5 years, it perfectly correlates. What do you do with the data to help?
Well, it simply becomes sort of an early warning system.
That's
I don't
correct.
think you necessarily can prevent anything, but at
No,
least you
you're
can
right.
anticipate it coming
You're right.
around and prepare for it.
I mean,
if
cholera
there's
has a
a problem.
similar type of scenario with regards to the coming of the monsoons, and then that comes back, and there is an early warning system based on the fact that you think you know why that's occurring.
Right.
But I think Vince is right in the sense that if it isn't testable, then it's going to remain conjecture.
Sure.
But it's pretty strong conjecture.
That's it's very convincing. It's
and
lovely data.
It's intriguing. It's
I
very
like it.
intriguing.
Yeah, I think this is another great example of medical ecology.
That's right.
That's right.
I totally agree with that. And but you need reinforcement of data, and I think that's a year after year. Based on this hypothesis, what's next year's pattern look like?
You should teach a course, Dickson, called medical ecology.
I already do that.
Oh, I saw you.
Thanks.
You just retired from doing that.
I did. I'm still in the last throes of this year's course, but this
Maybe you
will be the
should
last
put up a
one.
website or something.
I've got a website.
Oh.
Hey, you guys are ganging up on me here. Aren't
You know, Dick, if you have a website, you really ought to back that up on something.
Yeah, no, really.
Get a Drobo, yeah.
I mean, I thought about getting a policeman that could oversee that, calling it Drobo Cop, but of course that. Well,
That was a movie, right?
it was.
RoboCop.
Yes.
That's right.
Let's read a few emails. This one, Dickson, I saved for you. We had this queued up last week, but you weren't here, and I thought you would like it. Michael writes, "Hi, guys. Really like the show, even though some of it, not much, goes over my head. I also listen to TWiP and hope there will be a matching number of episodes to rival TWiV. My name is Michael, and I was talking to my dad the other day, and he mentioned that he took parasitology from Dr. Brown when he was at Columbia.
Wow.
Small world.
Wow. I was Dr. Brown's technician when I first began
Ask your
here.
dad, Michael, if he knew Dickson Despommier.
Cool.
Yeah, it was that guy always in the corner.
That's right.
Just
The
a
one that
sweet-
was asleep.
I was also interested in checking out some classes at UCLA. I live in LA and wanted to know if you had any recommendations as far as instructors for classes.
Go see Larry Simpson.
Larry Simpson.
Yep.
Is he a parasitologist?
He sure is. He got his degree at Rockefeller with William Traeger, and then later on went to
Okay.
work on leishmaniasis.
Well, if you're interested in virology, I know Ashim Dasgupta, Arnie Burke, Owen Whitty, and Sherry Morrison. She's an immunologist.
Right.
They would be great.
Yep.
And he continues, a couple of things. I was wondering how your iPhone app is coming along. I know lots of programmers and artists and would like to produce one of your ideas.
Wow.
It's not going anywhere. I don't have anyone to write it.
Should have one for isotherms.
What's your iPhone app?
I don't have one. I want to make one for viruses. I mean,
what do you want it to do?
well, I think it would just be something simple. You have all the viruses, and you tap one name, and then you go to pictures, information.
You can see whether it's one of the 7 different strains.
Yeah, you
of
could have
David
all the viruses in your pocket. So
yeah, it's sort of the virus database,
exactly.
the online database, only on your phone.
Right, but
Peterson's guide
that
to
would be the first iteration, and there would be others as well. But I think
it's going to be hundreds and hundreds of thousands of people that are going to want this app.
Oh, absolutely.
All right. It's not going to be that popular. I'm just trying to teach people. Right, it's a way of spreading knowledge. I don't know. I would have one.
I would have one. I'll buy it.
I know
Vince,
4 people.
Be careful. You might get stranded.
Hey, it would only be 99 cents to cover the programming costs.
All right.
I'd make it for the iPad, too, so it'd be a big version.
I'll bet you there's some way you can link it to one of the online databases.
Yeah, you'd link it. You'd get all the info, so you don't have to search. You just tap on polio. It's going to go to all the sources automatically.
Right. What's a tanapox?
Tanapox, yeah, exactly.
What is
I think there's a need for that.
I agree with you.
So anyway, he continues. Secondly, you guys mention all the time about the dearth of research being done. Well, we mentioned about the dearth of research money, not research.
Yes.
How would you like to start your own nonprofit? And is there a website that tracks current field studies or lists proposed studies? I have a great plan for a virtual corporation that could direct and help synergize field research. You guys would get to do the fun part, i.e. plan the protocols and general aim of the project and approve the personnel, take all the public credit. I would do what I do best, which is organization and logistics. I think Field Studies International sounds good, FSI.
Wow.
Well, enough of your time now. Thanks for the great podcasts.
Yeah.
Is there a website that tracks field studies? There must be, right, Dickson?
There's Google. Well, I think Google has medical maps of distribution of diseases and stuff, but nothing that's current.
Well, there's
the
the NIH site that or the government site that
used
tracks.
to be CRISP, right?
That's for
yeah,
grants,
that's
right.
right. That's right.
There's also one for clinical trials, clinicaltrials.gov, I think it's called. It's another one.
Don't know.
Nonprofit. Well, we have a nonprofit here. It's called TWiV, right?
That's right.
We don't make any profit.
We invert our pockets and nothing comes out.
Well, this one's from Mary, who has written to us before. She's a PhD student in Alabama, and she works on large DNA viruses, Rich.
Yep. I looked up her website.
Yeah, she works with Elliot Lefkowitz, who's a virologist there, who you probably know. Mm-hmm. As much as you guys are adamant against engaging in political discussion, I know you also feel strongly about exposing the truths behind alternative medicine, anti-vaccination campaigns, and so much other medical and scientific disinformation espoused by quacks all over the world. So maybe this is something you might feel like passing on to listeners. I'm not sure if it really counts as politics. Anyway, as you may know, science writer Simon Singh is being sued essentially for bringing debate on alternative medicine into the public forum, and the millionaire and corporate-backed British libel laws, among the most repressive in the world, are allowing this to happen to responsible journalists, writers, anyone even outside Britain, thanks to the thriving libel tourism the laws have given rise to. There is a movement sponsored by journalists and others, including scientists, to reform these laws, and it includes a petition for which they are inviting signatures from around the world, because even those of us outside the UK are at risk for litigation. So she sends us the links that you can see to sign this petition. Just food for thought. Please excuse me if this is wildly inappropriate. I don't think it's inappropriate.
No, I don't think so either.
This is a case I followed very closely as a science journalist.
And I agree with her. It's not politics. This is about truth.
Freedom of speech also.
Alan, is this still ongoing, the libel?
Well, the latest news, I think it was just yesterday, so in the latest appeal, Singh got the case dismissed, and then the British chiropractors decided they would not press it any further. However, Singh is out something like £200,000 of his money, or he's scraped up a few donations from places, I think. And so the remaining issue is whether the Chiropractors Association is going to be picking up his legal fees. And I think that's still up in the air. But
they sued him, right?
They sued him. So what happened was he wrote an article and about a chiropractic practices, and one of the things that he talked about was some of their claims about their treatments that are used pediatrically sometimes, and
Right.
and he essentially called these claims a load of hooey. And then the chiropractor sued him for saying this. In Britain, apparently, it is extremely easy to sue somebody for libel, and it is extremely difficult to defend yourself, which is why there have been a lot of these sorts of suits there. And this, in particular, was obviously very science-related, so I followed it kind of closely. But the upshot of it, I mean, if you go back to his original article and what the claims were, I don't see that. I don't think this would have gotten any traction in the U.S. It's really an issue of the way the British court system allows this to happen, I think.
So is there any standing provision in the British court system that says if you sue somebody for something like this, who pays for the legal fees if you lose?
My understanding of the British system is that you can, if somebody sues you and loses, they can be on the hook for picking up your legal fees.
Okay.
Which is, I would think, maybe a good thing about their system.
Absolutely.
That it would limit frivolous lawsuits. But in the case of their libel law is particularly, what would it be, plaintiff friendly.
And she continues, in a separate or maybe conceptually related vein of thought, here's a good essay from the Journal of Cell Science a year or so ago on the importance of admitting and even embracing our own stupidity as scientists. And this is by a fellow named Martin Schwartz. It's called The Importance of Stupidity in Scientific Research. And
it is excellent.
It's very good. I sent it out to all the students here the other day. So we'll post that. Thanks for that, Mary. And also, you have a very nice website. Maybe you'd like to put a link to TWiV on your website. Just a thought.
And
thought.
Vice versa.
Absolutely.
And it looks like I will see you in Sedona, Mary.
Sedona.
Sounds tough. That sounds tough.
Here, Dickson, you can see, this is Mary here. And she has all these links on the right. She should put TWiV, don't you think?
I think so.
Nice kids.
Yeah, couldn't hurt. Yeah, nice.
She says my personal projects here. Very good. Cedric writes, Dear this week's TWiV crew.
That's very political.
I heard in a lecture from Professor Stephen Goldman of Lehigh University that atmospheric wind currents can pick up dirt, bacteria, and viruses and transport them across entire oceans.
Correct.
Yes.
I was wondering if this can or could have been a means of viral spread. Can a virus survive in the upper atmosphere, or would radiation render it inert?
Oh.
Is this a phenomenon that can be of significance in today's world of human-created interconnectivity? What do you think?
Sure.
I think this predates human-created interconnectivity by a few billion years, actually. And there's a lot of very good evidence that microorganisms can survive pretty nicely in the upper atmosphere, up in the jet stream. They get picked up. I know there's been a lot of work on African dust storms.
Yes, that's right.
Which raise huge amounts of sediment, tens of thousands of feet into the air. And those then travel on the jet stream, and they show up in the United States.
You bet. And in China, too. They have a lot
and
of
in China, so there is a global stream of
microbial
stuff,
that's
microbial stuff that's just floating around. And you can isolate these things. You can go to high mountaintops and hold a sterile petri dish above your head for a few seconds and put the lid back on and grow all sorts of things that were floating around up there.
You bet.
I met a microbiologist who works for NASA, who his project is to try and figure out whether microbial spores could survive entry into Earth's atmosphere from outer space on meteoroids.
Wow.
Okay. And the answer is yes. Okay. So these are tough little guys, and they can get around.
Although viruses,
maybe even interplanetarially.
But viruses may have an issue with the UV. He's got a good point, right? Mm-hmm. They could be inactivated, their genomes, although some are always going to survive.
If they're included into a dust particle.
Exactly. In a dust particle, they would be protected.
They would.
Or in a bird.
Or in a bird.
Yes.
Birds don't go to the upper atmosphere, do they?
Well,
Not deliberately.
They try to fly over the Himalayas, and there was a wonderful segment on Planet Earth narrated by Sigourney Weaver, that showed a bunch of snow geese
Uh-huh.
that were trying to fly over the Himalayas, and they get about to the top of Mount Everest, and they can't breathe, so they have to come back down again, and then they have to catch their breath and go back up and try, and they have to get the right current in order to go across. But that's at what, 29,000 feet or something like that.
Cool.
Crazy.
Many thanks for your podcast. I'm a high school student working part-time as an intern in a national lab, and I love to listen to TWiV and TWiP while doing the menial chores.
There's no such thing as menial chores. They're all necessary, but you don't get paid big bucks for doing the small jobs, unfortunately.
Well, you learn a lot.
You learn a tremendous
amount. You
amount.
become pebbles and sand grains.
Well, you learn how the whole building's constructed. You can start from the foundation up, and I think that's a great start and a wonderful job to have, and I wish I had had one when I was a kid.
You did.
Not like that.
Well, not in high school. Later on.
Yeah, later on.
Salubrious salutations, he writes.
Wow.
Nice.
Who's that?
Cedric.
No, I meant who's the salubrious. He didn't show up for today's broadcast, I understand.
John writes, listening to your show reminds me of the excitement I felt as a child reading Paul de Kreef's Microbe Hunters.
Wow.
As a physician living in North Carolina, you are making me nostalgic for my days as a resident and intern at Columbia.
Wow.
Oh.
That's cool.
That is cool.
Your show reminds me of what a great institution it was and is. So thank you, John.
What was the year? We would like to know.
Tell us what year you were here.
Yeah, we'd love to know.
And we can drop some names of people who you might know.
Exactly.
But I'm glad you're listening in North Carolina. Take care of the people and spread the word.
Absolutely.
That's very cool.
Yep.
All right. This may be the last, maybe one more. Bernhard writes, you got me hooked. Cool. We're fishing.
Yeah, well.
Since I've discovered TWiV and TWiP, I'm addicted to virology and parasitism, well, at least during my commute to work and back home. It took me quite some time to catch up, but it was definitely worth it.
Wow.
Although I'm working as a software designer for medicinal chemists in a pharmaceutical company, I've always been interested in the big picture of virology. Listening to TWiV and TWiP as well gives me a glimpse into how biologists work and think, which helps me communicate with them. There are many other podcasts which I've since also subscribed to, but TWiV and TWiP are special in how they well match what I would be interested in from the respective fields and how entertaining the subject is presented.
Well.
Second reason I write is to suggest a pick of the week. You might know this. Last year I learned about Sustainable Energy Without the Hot Air by David McKay. No. I knew his previous book, Information Theory, Inference, and Learning Algorithms, as a treasure trove, but it's probably too heavy reading for general consumption. Sustainable Energy, however, is fairly easy reading, full of facts and figures, surprising calculations, provides a big picture, and shows how the world might be able to escape its current agony. It provides a sober view on British and worldwide energy consumption and production with real figures and personalized measures. It could arguably be the most important book on the topic written to date, so have a look and decide for yourself. Well, I'll check it out for sure.
Sure, I will too, of course.
It looks like a cool website.
Without,
I guess it's a website and a book.
oh, yeah, withouthotair.com. Sustainable Energy.
Oh, this is very interesting. Yeah, I guess they've got
look.
excerpts from the book on the website. It looks
Look
very
at
And
this.
you can download it as a free PDF
Oh, cool.
or
Yeah.
browse it as HTML.
Yeah, you can see all the different things making a difference at vertical farming here, food and farming. Let's check that out, Dickson.
knowledge for free, Vincent. How about that?
I love it.
Good.
I think it's great.
Knowledge for free is right.
I love milk. If I drink a pint of milk a day, what energy does that require? Oh. Hey, hang on. So 1 pint per day requires that I employ 1/32 of a cow. And then he goes on to calculate the energy cost. Keep up the good work, including the jokes. All right. And,
We started with one this time.
we did. Let's do one more. This is from Dave. Hi, Doctors VAD. Vince Allen Dickson forgot Rich.
That's okay.
Batter. When, of course, this is a long time
Well,
ago.
you know, it's a good thing I'm here.
When grid computing is promoted, the usual suspects mentioned are SETI at home and folding at home. At least of equal merit and more germane to TWiV should be the World Community Grid, WGG. So I think we picked folding at home as a pick of the week once. So he's promoting World Community Grid. harvests the unused computing capacity of volunteer PCs around the world, creating a virtual computer, and it's for scientists engaged in not-for-profit humanitarian research. And some of the research projects with a virology connection, muscular dystrophy, childhood cancer, help conquer cancer, human proteome folding, fight AIDS at home, influenza, antiviral drug research. If it would be good if you could give WCG an honorable mention. I'm an information technology professional with no formal training in any of the sciences, but I really wanted to make a contribution to scientific research, so my charitable cause is WCG. Since 2004, I have donated 88 CPU years to WCG products. This is the equivalent of turning a PC on in the year 1922 and having it run 24 days a week, 7 days a year, 24 hours a day, 7 days a week, 365 days a year, uninterrupted to the present day.
Wow.
And this, he says, this involves 10 multi-core PCs, 9 quads, and a dual for a total of 38 CPU cores running pretty much 100% load. Yes, my power bill does take a hit. Wow. As for TWiV and TWIP, thank you immensely for devoting your time and effort on the podcast, and especially for enthusiastically sharing your knowledge on virology and parasitism. I was hooked from episode 1 on both. I suspect I'm not your average listener type since I don't work in any scientific field and I'm circa Vincent's age. My career ship has sailed, but in my next incarnation, I hope I come back as a microbiologist or virologist. After finishing Volume 1 of Principles of Virology, it became obvious to me that to obtain a competent knowledge of viruses, I needed to take a deep plunge into biochemistry, cell molecular biology, and immunology. I'm just coming up for air, but what a fascinating journey. Science fiction pales in comparison to reality. Ready for Volume 2. You know, virology is a multidisciplinary science. You have to know a lot of things.
So is parasitology.
It is?
Yes. Sure, absolutely.
For me, I love that you don't dumb down the podcast too much. There is delectability in the details. For example, when you were talking about a membrane lipid, you use the actual terminology phosphatidylinositol. This is helpful so I can do further investigation and also very useful to hear how terms are pronounced. Mm-hmm. Having no contact with scientific types, I have to guess at how some terms are pronounced. This can be problematic since often online audio dictionaries will not have technical terms. Now, if we could just get Mark Pelletier from Futures in Biotech podcast to quit calling them amino acids, life would be good. So Mark Pelletier is our colleague, and I sent this email to Mark, and he said it's Canadian Insider Baseball, and he responded, my pronunciation of amino pays homage to a small town in BC just a little northeast of you. So Dave is from Victoria. I don't get any of that.
No, neither do I.
It's a Canadian thing.
Insider Baseball?
I didn't know they had baseball there.
They used to have 2 teams.
They have that ice game where they push rocks on the ice.
Now they have just 1 team. That's right.
Anyway, thanks, Dave. He says, I hope you continue to enjoy the podcast, doing the podcast as much as we, your audience, enjoy listening to them.
Yep.
And this site that he points out is really pretty cool. They use this, networking personal computers to do a lot of. They're actually computational biology problems, folding problems to look at the possibility of, for example, finding screening drugs that will bind to dengue proteins and that kind of stuff.
Mm-hmm. Yeah,
very interesting.
World Community Grid. We'll put a link to it for sure so people can look at it and think about giving their CPUs. And thanks, Dave, for your nice email.
Over and out.
And let's move on to a few science picks of the week. I'm going to start with you, Dickson.
Well, it may not be a big science pick. I actually emailed you an address.
And here it is, Dickson.
Yes, it's a general information on microbial diseases summary.
It's called Medical News Today, Infectious Diseases.
Yeah, it's called What's Up. In infectious diseases, that's what they probably
So they basically collect news on outbreaks and so forth.
So I don't know how reliable any of it is because I just discovered it, but I thought it was worth putting it up. But I also would like to recommend a book that just came out by a guy by the name of Bill McKibben. If anybody knows who that is. He wrote a book called The End of Nature. That's not the book I recommend. His new book is called EAARTH, to distinguish it from This Earth.
By Bill McKibben?
Bill McKibben. And it just hit the newsstands, and it's a fantastic book about what's wrong with the Earth and then how to go about interfacing and correcting some of those difficulties. So it's a general knowledge-based, big picture kind of a book that's I hope that a lot of people read it, and then you could use some of his recommendations to change public policy and this sort of thing.
All right. I noticed on this medical news today,
Yeah.
there's an article on polio eradication.
Indeed, there is.
Yes, which I had actually already put into the notes as an upcoming story.
Yep, we'll do
Bingo.
that. We'll do that next week.
Okay.
Yeah, there's some conversation about it.
There probably should be.
And Alan, I was talking about it with Saul yesterday, my colleague, and he reminded me of our article written years ago,
Yes.
which started the whole thing. But we'll talk about it next week.
Yes, we will.
Rich, what have you got?
This is as much an endorsement as anything else, because I don't know if everybody listening is going to be able to access it. But I'm a comics freak. I love the comics. Okay,
You and my wife.
and in our Sunday comics, at the very end, there's a panel called "You Can" by Beekman and Jacks, actually featuring Beekman and Jacks by a guy named Jock Church. And what this is, and I did find a link to a syndication site that gives you an idea what it looks like. Kids write in with questions, and this guy answers the questions and illustrates his answers, and a lot of them are technical and scientific questions, and he always comes up with little experiments that the kids can do at home
Cool.
to demonstrate the answers to his questions.
It's like Mr. Wizard.
Yes, exactly. And they're very, very well written, very entertaining. I like
neat.
the fact that he's taking an interest in
Yeah.
kids
Yep.
and having imaginative approaches and
Cool.
answering their questions and that kind of stuff. So if you can find it, if you've been skimming over it in your Sunday paper for the past several years, have a close look at it. It's well worthwhile. Show it to your kids.
Yeah, he
This
has,
latest one, I used to read this all the time when I got a paper that had comics in it. And this is really, really an excellent thing to have.
Nice,
yeah.
nice.
he's got 2 samples there.
And I love the
PDFs.
line in the first sample, gravity right now is a theory, say, because we don't actually know what causes it. But no one says just a theory when talking about gravity.
Right.
That's good. Yeah, it's cool.
That's
Yeah,
right.
gravity's a real poser, you know?
Yeah,
We really
right.
don't understand that, and it's pretty fundamental,
Here
but
we
it's
go.
just a theory.
It's
Yeah.
just a theory.
the theory.
That's right.
So actually, here's another situation where it would be nice if this knowledge were free. I can't find his strip online anywhere, okay? But obviously it's syndicated.
Right.
There's
like to be
there's
able to
contractual. Yeah.
Yeah, sure. But at any rate,
Cool.
good work,
Very nice.
Jock
Nice.
Church.
Thank
Keep it
you.
up.
Alan Dove from Western Massachusetts.
Yes, I have something I've been putting in my RSS reader for quite a while. It's this odd little news source called Underwater Times.
A little risque, Alan.
I just
Alan.
clicked on it today, and I just saw that there are 2 stories. That
What
Just
are we looking at
no,
here?
that is not the usual content.
Dixon is into this fishing story here.
I double fish. I see 2 fish here. Here's
Yes,
one
the
and
guy
here's one.
wading the fish. I mean, it's everything from people catching big fish to other things, but then pollution incidents.
Oh yeah.
A DNA analysis of the whale meat that was served in the restaurant in Los Angeles. Just news. Essentially, what the site does is it aggregates news from all over the web that is related to the oceans and waterways.
Right.
And as a scuba diver and boater, I am interested in this stuff. But it also gives you just a really interesting slant in a lot of cases on stories that you wouldn't have encountered, or you read other things about them, and thinking about them from the oceanic perspective is sometimes enlightening.
Interesting. Very interesting.
This is a very, very big hammerhead shark.
Yes.
Wow.
Australia. They have big sharks down there, don't they?
Right. It is.
That's right.
They do.
Ooh, that's a big guy.
It's very, very large.
That's pretty scary.
Yeah.
That's cool. Well, it's interesting that you pick a marine site, because my pick for the week is called the Reef Tank,
Oh.
which is all about the sea and fish tanks, marine fish tanks.
Huh.
And
Do you have a fish tank at home?
I used to. I don't any longer.
I did too.
They're a lot of work.
They are.
A lot of work.
Especially
yeah,
marine.
I used to have a lot
yeah.
of fish. I used to have cichlids from Lake Victoria in Africa. And that was a lot of fun. We had multiple tanks at once. I loved them.
Yeah.
But anyway, actually, a lady contacted me this week and wanted to repost one of my virus stories about viruses in the oceans. And she said it's for this site, and I came, and I really like it. It's got a gallery, a wiki, forums, and they get 250,000 visitors a month.
Interesting.
Product reviews, you're looking for an aquarium pump, you could find a review here. So anyway, if you like fish...
Oh, and here you are.
Here I
yes,
am.
here's your post.
Viruses of the sea. Okay.
Yeah. So if any listeners are coming over from the reef tank, welcome. And if you like fish, check out the reef tank. It's cool.
Great. Very nice.
And that'll do it for another TWiV. Gentlemen, Dickson Despommier can be found at verticalfarm.com now. I think you bought the new domain name, right?
It's org. It's actually org now. We bought the.
It used to be com. Now it's org.
Yes.
Medical ecology. Oh, before we go on, and your new book can be pre-ordered.
This is true.
The Vertical Farm.
That's right.
Make Dickson rich.
No, make St. Martin's Press rich.
Amazon.com or Barnes & Noble, you can pre-order. It's called The Vertical Farm?
It's called The Vertical Farm, The World Grows Up.
Well, if I read it, I'll make it a pick of the week if it's good. I'm sure it is because you're a fabulous writer. Verticalfarm.org.
Remember what Emerson said, so I remain speechless on this when he said, self-praise is no praise.
Mm-hmm. I'm
So
praising you.
I'm not going to say a word.
I'm praising you. And also medicalecology.com. Org also. And Tricanella.org.
Correct.
Media Empire.
Hey.
Good to see you again.
It's great to be seen.
Alan Dove is at dovedocs.com. Thanks for joining us again.
I do not have a media empire, just an umpire.
Nice to hear your voice.
You are the
Alan.
media. You are the media.
Yes,
That's right.
I am the media.
The media is the message.
And your knowledge of all things media related is very nice to have here on TWiV.
No question.
He knows a lot of things that we certainly don't know.
Absolutely.
You think about all kinds of science. That's what we don't. Well, I don't anyway. I think about viruses. And from North Central Florida.
You got it.
Thanks for joining us, Rich.
Good to be here.
Always nice to have everyone here together.
That's good fun.
That's Rich with the Wallace Beery-like voice.
You think that's Wallace Beery-like?
It is Wallace Beery-like. Don't you think you have a Wallace Beery-like voice?
I don't even know who that is.
Oh, you should check him out. He's a wonderful actor, and had a gruff, tough, kind of a voice about him, but it was a very gentle man. As I'm sure you are too.
Wasn't he a star of silent films, Dickson?
Yeah, but he transitioned into the spoken word versions.
Oh, he did, because otherwise you wouldn't
Wallace
know
Beery.
that his voice sounded like Rich Condit.
This is all true. But
well, everyone, thanks for sending your questions. As usual, send them to TWiV at twiv.tv. Questions, comments, corrections, anything. We love it. Audience involvement is terrific.
Yep.
Thanks, everyone. Thanks for listening. You've been listening, of course, to This Week in Virology, which is all about viruses. We'll be back next week. Another TWiV is viral.